Yeah, so I'll be presenting Grim filter, which is a fast seed location filtering mechanism in DNA read mapping, which uses processing and memory technologies. And this was published at this was presented at APBC, and published in BMC Genomics back in 2018. So I'll begin with an executive summary. Genome read mapping is a very important problem and is the first step in genome analysis. Read mapping is an approximate string matching problem, and it's essentially for the problem that we're particularly looking at, we are trying to find the best fit of 100 character strings into a 3 billion character dictionary, and that 3 billion character dictionary is the human genome. So alignment is currently the best method for determining the similarity between two strings, but it's very expensive as it relies on a n-squared dynamic programming algorithm. And so we propose an algorithm called Grim filter, which accelerates the read mapping process by reducing the number of required alignments. And we design Grim filter such that it's able to be easily accelerated using processing in memory, which is a technology that basically adds simple logic into a 3D stack memory. And it has high internal memory bandwidth and enables performing parallel filtering. So with processing in memory, Grim filter is able to deliver up to a 3.7x speedup. I'll first begin with the motivation and goal. And this is basically the outline of the talk that I'll be giving. So sequencing refers to the process of determining the series of nucleotide base pairs in a DNA strand. And DNA is comprised of 4 different base pairs, which are referred to as A, C, G, and T. Today's machines sequence short strands, which are referred to as reads, and reads are on the order of 100 to 20K base pairs. I guess nowadays they can be longer, but that was what it was back in the day. As a general rule of thumb, sequencers that result in larger reads are more expensive and produce more erroneous reads, whereas the ones that produce shorter base pairs are cheaper and are more accurate. And so that's why we're basically focusing on these shorter reads in our paper. And the human genome is approximately 3 billion base pairs. Therefore, the genomes must be cut into reads, which are sequenced independently and then reconstructed. So read mapping is the first step in analyzing someone's genome to detect predispositions to diseases, personalized medicine, and of course, popularly now is to identify different COVID variants. So the goal here is to accelerate end-to-end performance of read mapping. I'll go into the background of read mappers, and in this paper, we focused on hash table-based read mappers because they were more accurate and more comprehensive compared to other methods. So what read mappers do is basically take a number of the sequenced reads, which are around 100 base pairs in our paper here, and we reconstruct the full genome, which is the green strand on the right. And so thankfully, we have a known reference genome that exhibits 99.9% similarity across humans. And this is generally true across any organisms of the same species. And this reference genome enables us to map our reads to the known reference with some minor differences allowed, which then requires the approximate string matching problem. And the differences between like my genome and the reference genome or someone else's genome is what makes each of us unique. So because the reference genome exhibits high similarity, long sequences in reads match to the reference genome perfectly. So here we have 9 sequential nucleotides in this read, this red strand read here of size 11 that match perfectly to the reference genome. So we can use hash tables to quickly help map the reads. And I'll talk about how we do that next. So before mapping any reads, we generate a hash table once per reference, which can be used to map any number of reads to the specific reference genome. The keys in the hash tables are k-length sequences, which are referred to as k-mers, and the values are lists of locations in the reference genome that the particular k-mer occurs in. So in this example, we see that the sequences of 5 A's here on the left occurs in 6 locations, which is shown on the right. We generate the key value pairs for every permutation of k-mers for the chosen value of k. And so this hash table depends on, the number of keys depends on the size of the k-mers chosen. And so here, just to emphasize, we see that the sequence, the k-mer AAAAT, occurs starting at the 36th and at the 434th base pairs of the reference genome. And so if you see that sequence, then you can immediately go to that location in the reference and then extract the sequence of relevance. So we can query this hash table to get the substrings from the reference and basically localize our approximate string matching to those regions rather than looking at the entire reference. And so this hash table based method makes things very fast. So here I'll show you the process of how we can actually use this hash table in read mapping for a single k-mer and a single read. Note that for every single read, we must extract x plus 1 k-mers, where x is equal to the maximum number of allowable errors between a string match. And this essentially guarantees that one of the k-mers will match perfectly in the reference genome. And this comes from the pigeonhole principle, if you're familiar with that. So from any given read, we select k-mers and retrieve their location lists from the hash tables. And then we retrieve the appropriate reference substrings from the reference genome according to the location and the offset of the k-mers within the read. We then compare each of these reference substrings against the read sequence using alignment, an expensive dynamic programming algorithm. And we note that cases with clear mismatches should not waste compute resources via string comparison. We also have to repeat this entire process for many, many reads whose order can be approximated by dividing the genome length by the read sequence length. In the case that the human genome is length 3 billion, and for 100 base pair read length sequences, we will have at least 30 million reads. And this will increase as you want higher coverage of the references, which will give you higher confidence of your alignments. So most of these locations we compare do not match the reads. We find that 99.9% of these actually result in a mismatch. So it's more efficient to build a filter that quickly identifies and discards such locations without doing the comparison to the reference genome. And so we want to filter out these locations that will result in mismatches very quickly. So alignment is the algorithm for determining a match between a read and a reference substring. And this is very expensive and requires the use of the dynamic programming algorithm. To give a scale of required computation, a single human can generate millions to billions of reads, and each read results in multiple locations. So you'll have to check many of these locations. Modern read mappers reduce the time spent on alignment for increased performance, and this can be done in 2 ways: by further optimizing the alignment algorithm, which many prior works have tried, or by reducing the number of alignments necessary by filtering out mismatches quickly. Both of these methods are used by mappers today, but now filtering has replaced alignment as the bottleneck. So our goal is to improve the filtering step in read mappers. So now let's take a look at how the hash table-based read mapper would work with a filter. So taking the same read split into k-mers of length 5, and we query the hash table for the location lists. We then pass the locations through the filter and attempt to efficiently discard as many locations as possible before having to run alignment. Here we see that the locations that are obviously mismatched get discarded before needing alignment. And then all the sequences that pass the filter must be aligned against the string, against the original read. So any location that passes through the filter but then fails the alignment is called a false negative. And false negative rates and the filtering speed are important metrics for analyzing the filter. So note that when the filter was added, we were able to forego 3 instances of alignment here, thus saving us execution time. So now let's get into our proposal, which is grimfilter. I'll talk about grimfilter in 3 steps. First, we'll discuss the data structures required for grimfilter. Then we'll talk about how we query the data structures, and then we'll talk about how we integrate grimfilter into a mapper. So the data structure necessary, we define as bins, and we basically take the full genome and partition it into these large sequences called bins. So we have a bin here that represents the sequence, and then we we create a bit vector that represents a bin, and the bit vector essentially is a is a array that holds the occurrence of all permutations of a small string or a token in the bin. So here, in this particular bin, this bit vector indicates that there is a small token of 5 A's here, but then there is no occurrence of 4 A's and then a C here, based on the 1 and 0 values of this particular bin. Yeah, and then to account for, and because we want like a whole read to occur within a single bin, we have to chunk the reference into multiple bins that overlap. And so basically, any read is guaranteed to fall within fully fall within a given bin. So this overlap basically has to be at least as long as the size of the read.
Jeremy?
Yes.
How we decide on the size of the bin itself and the token?
Yeah, so obviously there's more information if you have like less populated bit vectors. And so you can like extract more information. Obviously, if the bin's too large, the bit vector will likely be like all ones. And so you kind of want to tune the bin size and the token size to have like a reasonable rate of ones and zeros. We don't really have a, we basically just like sweeped a bunch of different sizes and heuristically found specific values that were good for the human reference. But obviously this can be tuned based on whatever particular reference you're using.
I see. And do you consider the reverse complement of the strand or just the forward strand?
Yeah, that's a good question. So we only look at the full string. But yeah, I guess in hindsight, we probably should have been looking at the reverse complement as well, because those are also valid alignments.
I see.
Yeah.
Do you think you should build another bit vector to consider the complement of the strand, or maybe you can find a simple hack?
Yeah, so I would say the easiest way to adjust that is to, for every single read, basically just reverse complement the read and then map it as well. And then it should work.
So, I see. Thanks. Yeah, for all other students, feel free to ask questions in the chat or just enable your microphone. Go ahead, Jeremy. Thanks.
Yep. Thanks. So let's look at, in a closer way, how these bit vectors split up the reference genome. So we have these bins that are overlapping across the whole reference. And we have these bit vectors that represent each bin here. And so basically we have a lot of these bit vectors depending on like the bin size, and obviously the bit vectors depend on the token size. And so to give you a feel of how many bit vectors or how large the memory consumption would be just for this, we can calculate it by using like the number of bins that we split the reference into, and we multiply that by the size of the token or the size of the bit vectors. So this equation is just like 4 to the n, because there's 4 different sequence types or nucleotides, and then t would be the number of bins. And so for a bin size of around 200 and a token size of 5, the memory footprint is around 3.8 gigabytes, which is reasonable for modern memory sizes. So it can completely fit within like these memory constraints. So now let's talk about how we would go about checking a bin for existence or possible existence. So we have the read sequence here, and we first try to extract all the tokens from the read sequence. So this is all the overlapping tokens here of size 5. So we see that we're just extracting them. And then we basically want to see how many of these tokens exist within a bin of a particular location in the reference. And if you think about it, like more occurrences of tokens that are shared between a read and a bin indicate higher likelihood of that read existing within the bin, or that read matching well within the bin. Yeah, so if there's like an error here, maybe a couple of these tokens will be missing, but depending on, I mean, if everything else is there, then for the most part, you'll have a high number of token matches. So taking all these tokens, we look at the bit vector for a given bin number, and we query all the bit vectors with the token indices according to the bit vector setup. And we basically just sum all the existence bits here. Afterwards, we compare to a threshold, and if it's below the threshold, then we discard it because we don't have enough matching tokens between the read and the bin. But if it's above the threshold, then we can send it to the read mapper for sequence alignment. And this is basically how we're discarding potential locations that must be aligned. So how would we integrate GrimFilter into a full read mapper? So GrimFilter takes in a read sequence and all the potential seed locations as input. And remember, these potential seed locations are taken from the output of querying the hash table. And it feeds it to the GrimFilter, which essentially calculates the existence or the potential existence of that read sequence within every single bin of the potential seed locations. And so it'll spit out a seed location filter bit mask, where 1 indicates, yes, you have to check this bin for existence, and 0 indicates, no, it's definitely not there because there weren't enough tokens. So we send this bit mask over to a seed location checker, which compares the seed location checker to the potential seed locations, and only passes on the seed locations that actually pass this seed location checker, which is basically the indices that have a 1. Anyone that has a 0 is basically discarded. We then send these locations to the reference segment storage, which extracts the segments from the reference. And then we finally calculate the edit distance using the read mapper. And so here we see that we're discarding 1 out of 3 locations of the visible ones here, at least, and therefore we're saving time on the actual edit distance calculation. And then we output the correct mappings. So how do we map GrimFilter to 3D stacked memory? So as a recap, GrimFilter is comprised of very simple addition and comparison operations. And to check a given bin, we sum the bits corresponding to each token in the read. And then we compare the sum against the threshold to determine whether to align. So it's highly parallel. Each of the bins can be operated on independently, and there are many, many bins. And it's also memory bound because we have to frequently access the large bit vectors we have to access them very frequently to calculate the potential existence. And these 3 properties we find together make GrimFilter a good algorithm to be run in 3D stacked DRAM, which has high throughput and like you can operate on things very parallel inside the logic layer. So 3D stacked memory is basically a bunch of DRAM layers that are stacked on top of each other. And they're connected with TSVs, which are just vertical connections that allow high bandwidth between layers. We also have a logic layer, which enables you to move DRAM data at high bandwidth between the layers to the logic layer, perform calculations, very simple calculation on the logic layer, and then put them back into DRAM, depending on whatever is implemented in the logic layer. And so we have this high bandwidth as well as this stacked customizable logic layer, which enables the processing in memory. And this processing in memory offloads computation to this layer rather than transferring all this memory or data into the CPU, having the CPU process it and bringing it back. So it alleviates the memory bus. And we can embed these GrimFilter operations into the DRAM logic layer and appropriately distribute the bit vectors throughout the DRAM layers so that we can effectively parallelize these operations in the logic layer. And there's a lot of these 3D stack DRAM technologies that are commercially available, such as high bandwidth memory and HMC. So each of these DRAM layers are organized into arrays of banks, and a bank is an array of cells with a row buffer. So the row buffer holds one row at a time and uses this structure to transfer data with high bandwidth into the logic layer via TSVs. We now show that the distribution of bit vectors across the DRAM arrays to enable parallel processing of the bins. So each row represents a token's occurrence across many sequential bins, and the row of the appropriate tokens gets queried to find a vector showing the occurrence of the token across many bins. And these are the vectors that are brought down into the logic layer to be operated on. So now let's look at the implementation of the logic in the logic layer. Grim-filter only requires very simple design for enabling bit vector sum and threshold compare across many bins simultaneously. One comparator, one incrementer, one accumulator, and a buffer for each of the bins that we are checking in parallel. And this results in very low overhead and ease of implementation. So for example, in HBM2, we use 4,000 incrementer LUTs, 7-bit counters and comparators in the logic layer, which is very simple to implement. And of course, there are details in the paper. So now let's get into the results. So to gather our results, we did performance simulation using an in-house 3D stack DRAM simulator. And we evaluate 10 real read data sets, which is from the 1,000 Genomes project. Each data set consists of 4 million reads of length 100. And we evaluate 2 key metrics, which is performance and the false negative rate, which, if you remember, is the ones that pass the filter but do not pass the alignment. And we compare this against the state-of-the-art filter FastHash, which was the state-of-the-art at the time, when using Mr. Fast. But Grim-filter can be used with any read mapper. So here we have the plot of performance, where the y-axis shows the time in terms of 1,000 seconds each. And on the x-axis, we show the different data sets. This is just how we label the data sets in bioinformatics. And we plot for both the FastHash filter and for Grim-filter for total execution time. And we use the sequence alignment error tolerance of 5%, which is pretty typical. So we find that we get a performance benefit of up to 3.7x here, and on average 2.1x. And Grim-filter gets a lot of performance due to its hardware and software co-design. So in terms of the false negative rates, where the false negative rates here are on the y-axis, we get 6x, up to 6x fewer false negatives, which means that you can check much fewer locations for alignment. And Grim-filter utilizes more information available in the read to filter because we're extracting overlapping tokens, whereas a lot of places don't use the overlapping information, and so throw out a lot of information that might be there. There are a bunch of other results in the paper, such as sensitivity of execution time and false negative rates to error tolerance of string matching. We look at the read mapper execution time breakdown, and we look at sensitivity studies on the filter for the different parameters like token size, bin size, and error tolerance. And in conclusion, we propose an in-memory filtering algorithm to accelerate end-to-end read mapping by reducing the number of required alignments. The key idea is that we introduce a new representation of coarse-grained segments of the reference genome, and we use massively parallel in-memory operations to identify read presence within each coarse-grained segment. Our key contributions and results are that we have a customized filtering algorithm for 3D stack DRAM, and compared to the previous best filter, we observe up to 3.7x read mapping speedup, and we have up to 6.4x fewer false negatives. And Grim-filter is a universal filter that can be applied to any read mapper. So again, here's the slide that shows the title, and you can access the paper through all these links or QR codes. And yeah, thank you for your time.